MC, my colleague Chris Rhodes, is going to come up and introduce Pascal. So Chris, could you take over, please?
So welcome to the afternoon session for the showcase. The first speaker up, we have 5 speakers. The first speaker is Pascal de Châtelet from Apellis, who's going to tell us about the complement inhibitor program, I believe, that they have going on there. So let's have at it, Pascal.
Good afternoon. Well, I would like to thank the organizers for a chance to speak and present some of what we've been doing. I'm with a small company called Apellis. Essentially, we're a small virtual company. There's 6 of us. We're basically born out of a previous company called Potentia Pharmaceuticals that was essentially acquired by Alcon in 2009. Now Novartis. We were developing a peptide for age-related macular degeneration, and we brought it up to the end of phase 1. Alcon picked up the program, but we're able to retain the rights to all the peptide family for any indication outside of the eye. So we decided to form a new company around this opportunity and basically look at new indication. So we have 2 main programs. The first one is developing a peptide for severe asthma COPD. So it's well designed for pulmonary delivery. It's stable, can be nebulized. It gets clear quickly through systemic circulation. And the second program is actually to extend the half-life of the peptide to allow some IV sub-Q systemic dosing to allow targeting systemic and chronic indications. So first, the peptide that we're developing is inhibiting the complement system. What's the complement system? It's part of the innate immunity of the body. It's a very complex system. It's got 3 initiating pathways, a classical lectin alternative. What's really important there, they all basically merge at the level of C3, which is the major protein in the pathway. And basically inhibiting at C3 like we're doing basically shuts down the entire pathway. It's obviously a pathway that protects you against pathogens, but as we get older, in some cases get activated needlessly and actually leads to tissue damage. People recognized that this was an interesting pathway to target for drug development, except there was no real good drugs to target the pathway until the advent of biotech. And the first drug to target this pathway was approved in 2007. It's Celeris by a company called Alexion. Right now, it's the most expensive drug in the world. It's been a blockbuster. It's going to reach $1 billion a year sales this year. So there's a long list of actually disease where the complement system is believed to play a major role in the pathology of the disease. And we believe actually that more than just the complement being involved, we think there's a lot of this disease, Th17 disease, and a lot of disease like asthma, COPD, psoriasis, rheumatoid arthritis, actually are probably some kind of autoimmune disease, not like large systemic autoimmune disease like lupus, but something more like tissue-specific autoimmunity where you have kind of a cycle that gets kicked in. You have an acute injury or insult that can be oxidative damage, allergen exposure, or infection. Basically kicks the system into a loop, the local system into a loop where you basically have activation of the dendritic cell in the tissue, which basically drive Th17 polarization of T cells, which basically gets B cell to produce a lot of low affinity monoclonal antibodies, which either kill the cells themselves or activate complement. Complement activation both will continue to activate dendritic cells or actually activate phagocytes who generate more tissue damage. So there's this kind of cycle that the tissue gets kicked in that the body can't really get out of. So we believe the difference between macular degeneration, rheumatoid arthritis, psoriasis, it's basically the same cycle, just happens as different tissues. And we believe that by inhibiting complement, you can actually break the cycle and allow the immune system to basically go back to quiescent state. So essentially our peptide is APL1. It's actually part of a family of C3 inhibitory peptide. And how it came to be, it's basically been discovered in the 1990s by a professor called John Lambris at University of Pennsylvania through phage display. So this is a purely synthetic peptide. There's no biological homologs. Basically, it was screening the library against C3, identified a consensus sequence, 13 amino acid cyclic peptide. After a bit of work, found out that it was very specific for primate C3, so it works well against cynos and rhesus, but if you try to do any work in mouse or pig, there's no activity. Also discovered that if you cap both ends, acetylation and amidation, you end up with something that's very stable in the serum also. Mechanism of action is pretty well understood now. There's even a crystal structure of the peptide bound to its, one of its receptor. It's not a receptor, but it's binding pockets, let's call it. And basically the way it seems to work is normally C3 is a big molecule. It docks with the enzyme complex and gets cleaved in the C3b and C3a fragment. Comstatin and other peptides of the family probably act as a little stone in the gear, just prevents proper docking with the enzyme system and basically just stops the cascade at the C3 level. APL1 is a third generation derivative. It's been optimized through rational design and library screening. It's got by SPR binding affinities between 100 and 500 picomolar. So at this point, we're talking comparable to many monoclonal antibodies. It differs by the original constant by 2 amino acids, one of them being an unnatural amino acid. We've tested they actually inhibits both pathways, well, 2 of the 3 pathways of complement inhibition, so it's active as you expect. And as I've said also, this has been in men before. It's been for treatment age-related macular degeneration. We ran a phase 1 in 27 patients, actually with people with very advanced stage of disease that are essentially blind. And people with wet AMD, what you're seeing there is kind of a side view of the retina. People with wet AMD has got exudation in the retina, so you see this kind of blister, this pocket, the fluid-filled pocket there. That's bad. And we gave a single injection in the vitreous humor of the patient. It forms a deposit there, and you can see after 1 week, nothing much is happening. And the peptide's got a half-life about 1 day, so about a week it's basically gone from the eye. And then if you go at 1 month, it fully resolves after a month. So we notice there's definitely something that's happening after the peptide's gone, after the pharmacology is out. There's something fundamental that changed in the back of the eye. And we believe essentially we're inhibiting the cycle, we're just breaking the cycle and allows the system to resolve. And there's some history of complement inhibition in asthma and COPD. So first, asthma, COPD, big unmet needs, severe asthma and COPD. The only thing right now for asthma is bronchodilators and steroids. Severe asthmatics are not responsive. COPD patients are not responsive, so huge market. More than 10 million people with billions of dollars potential market. And one of the reasons we decided to focus on asthma initially was first because the peptide was well suited for pulmonary delivery, but also there's been a study with this other drug. It's an anti-C5 antibody that's been approved. So Soliris is an approved drug. It's been tested in asthma before. In this model, in patients, mild asthmatics, been done in Canada, where basically you challenge them, you have them breathe in allergens, which basically stimulate a mild asthmatic attacks, and you can track lung function. So essentially the way the trial works is you have a first challenge where you establish baseline hyperresponsiveness, then you basically dose with the drug. A day later, you do another challenge, and you see how the drug interfere with the asthmatic response. And then you wait 28 days where the drug actually washes out of the body, and then you challenge again to show that it's back to baseline. This is part of crossover design where you would normally then use the same patient with placebo as a kind of crossover. And the data was pretty interesting. Here is the forced expiratory volume table. The lower the curve, the worse it is. So you can see the blue line is your baseline. That's a standard asthmatic attack. And 1 day after the drug was administered, you can see the patient do a bit better, about 30% reduction, which is good but not great. Steroids will give you 50 to 60%. And then 20 days later, after the drug has washed out, again, you see 60% reduction instead of going back to baseline. So the same kind of behavior that we see way beyond the direct pharmacology of the drug and impact in the lung environment that makes it less asthmatic, more reacting like normal lungs. So we repeated this study design, but with cyno. So we're getting ready to start our first clinical trial already next year. But we run an animal model where you basically take cynos are allergic to a small parasite called the ascarisum. You have the primates actually breathe in the dust of the sum, and they basically generate an allergic response in the lung. And you can measure various reactions to the allergen, and the cytokines are usually the most telling. So the design was basically 3 groups, one receiving the drug for 14 days on a daily dose. The second one control the 31-budesonide, which is basically state-of-the-art steroid. And as expected at challenge 1, steroid performs beautifully. APL-1 also performs almost as well as steroids in all parameters. And interestingly, 28 days later, after all the drugs have washed out, we get basically the same impact, the same reduction of cytokine upregulation in challenge 2 than we do in challenge 1, basically repeating what's been observed with Celeris previously. And at this point, we're getting ready, I said, we finish IND-enabling toxing, ready to get in men. And then we have our second program. We're basically extending the half-life. This is a peptide, so it's got a short half-life. Terminal half-life about 6 to 8 hours, which is not bad for peptide, but not good enough if you want to do any kind of chronic indications. And essentially, standard strategy, you take the peptide, you attach something big to it or something that allows it to stay in the blood longer. So we found a good linker, and we look at a number of moieties, and this is basically the data we observe. And the dark red line is the naked peptide, which, as I said, is going to half-life about 6 to 8 hours. And depending on the moiety that we use, we're able to get about 5 days of half-life. Even sub-Q, we've tested some daily sub-Q doses, and we can get, we believe, what would be a nice therapeutic dose in men. So we believe sub-Q then becomes possible. We can do systemic complement inhibition and target a lot of the systemic diseases out there, including all the diseases that Celeris right now has been approved for, that we think we can probably provide a superior alternative. And that's basically in conclusion, we believe complement inhibition is a way potentially of resolving, not trying to suppress the immune system to release symptoms of the disease, but trying to do the immune correction where you break a cycle of autoimmunity and allow the system to actually calm down. And we think we might just need to have a cycle of immune correction for 2 weeks and basically just resolve a lot of these autoimmunity like psoriasis, rheumatoid arthritis, multiple sclerosis. That's the dream, obviously. But then have the immune system calm down and maybe just retreat every few months when the symptoms or the autoimmunity comes back. So APL-1's been shown that it can be delivered through the lung, it's active, it's got promising properties, so moving that forward. And we also got good preliminary data with APL-2 as an alternative to basically address chronic inflammatory disease for actually a very wide range of potential indication moving forward. And thank you for your time.
Complement system has beneficial effects. How do you address the inhibition and how that affects the beneficial effects of the immune system?
Well, that's an ongoing question. And that's partially been answered by Celeris. Celeris is people getting inhibited at C5 level, and many of these patients have been on the drug for 2 years. And beyond some kind of susceptibility to meningitis, which you can vaccinate against, they actually do very well. But that's an ongoing question, is obviously the complement system does something. So if you do inhibition, you might do some kind of immunosuppression, which you have to guard against. But there's a lot of immunosuppressive regimen that are emerging, Humira, I love these things. So I think something can be managed. So far, we've not seen, all the work we've done in Sino, both in the eye and IV, we've not seen any sign that these animals become very severely immunosuppressed. The other point, there's a lot of people walking around our natural genetic C3 deficient individuals, and they're not like totally immunosuppressed. They walk around, they just have to be careful with some type of meningitis is a big one, but otherwise they're healthy individuals.
Pascal, I have a question about the intended or expected human dose for the asthma indication. In the cynos, that was a pretty high dose. How do you calculate what you expect the dose to be in humans?
That's going to be probably the highest related dose is what we move for. There's no good animal model, so it's very hard to know the dose that we need. We have the animal model, gives us an idea, and that was 10 mg per kg. I think probably my guess is a mg per kg or they are where you need to be. But we'll know when you go in the clinic. For sure, there's no good animal models or precise way of establishing that.
Right. Thank you. So are there any other questions for Pascal? Okay, if not, let's thank Pascal again.